We've heard a lot of discussion about changes at the level of the DNA in cancer cells and changes in how, changes that affect the transcription of the DNA into RNA, but there are also changes in how the RNA is processed and how the RNA is stabilized or destabilized, and whether it's able to be translated. And the last presenter of the day is Dr. Chonghui Cheng from the Department of, from the Division of Hematology Oncology. She's an assistant professor there, and she devotes most of her laboratory to studying changes in RNA splicing that occur in cancer. So she's going to talk to us about post-transcriptional regulation of gene expression and how it's altered in cancer. Thank you.
Thank you for staying so late. So I'm going to keep it brief. If you have any questions during my talk, raise your hand. So today I'm going to talk to you about post-transcriptional regulation. And there are two major post-transcriptional regulations that occur during the level, at the level of alternative splicing, as well as at the level of microRNA regulation. And there are other post-transcriptional regulations, such as mRNA degradation through nonsense-mediated mRNA degradation that I'm not going to cover this time. So in the eukaryotic kingdom, there are multiple different organisms, from the single-cell organisms such as yeast, to mammals like us that contains hundreds of cell types, tissues, organs, and systems. So showing here are different cell types that are composed by mammals. However, if we are looking at the numbers of genes, we find that in single-cell organisms such as yeast, it has 5,000 genes. And we humans have no more than 25,000 genes, no more than 5 times as the numbers as compared to that of the single-cell organism yeast. So what that means is that the number of genes cannot really explain the complexity of humans that we are as compared to the less complex organisms such as yeast. Other levels of complexity must exist. And if we are looking at how a protein is made, which contributes to the diversity of functions in a cell, we know that, I think I'm not so good at using these buttons. We know that DNA is transcribed to pre-mRNA. And in the nucleus, pre-mRNA is spliced to make mRNA, mature RNA. And this mature RNA is polyadenylated, which is exported to cytoplasm where translation occurs. However, if we are looking at mammals, what happens is that going from DNA to pre-mRNA, it's pretty much the same as single-cell organism yeast. However, as you just have heard, there are multiple modifications that can occur in this process. In addition, these pre-mRNAs can make multiple different types of mRNAs with the inclusion or exclusion of different of the exons, as shown here. So for those who do not know, pre-mRNAs are composed of introns, as I shown here in thin lines, and exons shown here as boxes. So exons are the sequences that encode proteins. However, introns are spliced out to make mRNA, so introns do not contain any of the coding sequences. So in this case, mRNAs are made by including or excluding different exons, resulting in different mRNA species further that are translated into different proteins. So in single-cell organism yeast, generally it's one DNA, one protein. However, in mammals, one DNA can make multiple mRNAs that result in multiple protein isoforms. And these protein isoforms often have different functions in cells. And this process is called alternative splicing. So just to emphasize this process again, DNA, so in single-cell organism, DNA is transcribed to pre-mRNA, and pre-mRNA is spliced to mRNA, and mRNA further is translated into protein. However, in higher organisms, such as mammals, alternative splicing occurs, resulting in generation of multiple mRNA species, and that will be able to make multiple protein isoforms from a single DNA. Now, this process, in fact, in humans is quite prevalent. It has been shown that the majority of human genes undergo alternative splicing. And recently, bioinformatic analysis shows that about 92 to 94% of our genes undergo alternative splicing. So for the gene names that you know, most likely that gene undergoes alternative splicing. And one of the major alternative splicing events is shown here, which means that these two exons shown in gray are called constitutive exons. So in every single event, these constitutive exons will be included into the final messages. And in the middle, the magenta one is called variable exons, or alternatively spliced exons. And this exon can be included into the final message called exon inclusion, or it can be skipped so that it's called exon skipping. Yes?
Is this pure chance, or is also this regulated, whether it will be?
Yes. So the question is whether this is by chance or this is by regulation, and I'll get to that. This is a tightly regulated event. So not only prevalent, alternative splicing is also versatile. It can be used as a switch. For example, the Fas receptor protein is an apoptosis protein that you just have heard this afternoon for apoptosis. So cells die by apoptotic process. What happens is that the Fas receptor undergoes alternative splicing. Inclusion of this variable 6 exon results in a membrane bound form of Fas that promotes apoptosis. However, exclusion of this variable 6 exon results in a soluble form of Fas in which it inhibits apoptosis. So by regulating the alternative splicing of the Fas gene, cells can control whether they want to undergo apoptosis or they want to survive. Alternative splicing is also a fine tuner. For example, the myosin heavy chain gene can undergo alternative splicing that generates multiple different isoforms of the myosin protein, which controls the slow and fast muscle contraction, which really facilitates us to move or to do anything that we want. Alternative splicing is also a diversity generator. For example, the Drosophila dsCAM gene is an extreme example. This single gene can provide up to 38,000 mRNAs, and it is the isoform of the dsCAM gene or dsCAM protein that wears up the fly nervous system. So you can envision that dysregulated alternative splicing can result into diseases such as cancer. And in fact, it has been estimated that about 20 to 30% disease-causing mutations in humans affects pre-mRNA splicing. And this is an underestimate because it really doesn't include variations that are contributed by splicing factors or factors that control alternative splicing. So from this effect, you can think that alternative splicing really needs to be tightly regulated. So showing here is a signaling network where each of the signaling cascades is important in terms of controlling some of the processes in cells so that our cells can function properly. today what I'm going to tell you a little bit is one of the pathway called Ras-MAP kinase signaling pathway, in which receptor tyrosine kinase on the cell surface is activated by extracellular cues such as growth factors. And this activation results in activation of Ras-MAP kinase signaling, leading to activation of RAS, RAF, MAC, and MAP kinase. And many of these genes undergo alternative splicing. For example, many types of receptor tyrosine kinases, or RAS, are known to undergo alternative splicing. In addition, many of you must have heard this p53. And p53 is also undergoing alternative splicing. And it is known that different isoforms of p53 performs different roles in cells. Other examples includes FAS, as I just pointed to you, RAF in the Ras-MAP kinase signaling. SRC is another kinase that's quite important in terms of controlling cellular processes. So again, it indicates that alternative splicing is quite important in terms of controlling cellular processes, and that dysregulation of this process results in diseases. Now, the major fundamental questions regarding alternative splicing are: what signals control alternative splicing? And what are the consequences of alternative splicing that's regulated by signaling that leads to diseases such as tumor genesis? And our lab uses CD44 as a model system to study these fundamental questions. So today what I'm going to do next is to just use CD44 as a model system to show you how important this regulated process is. So CD44 is a cellular membrane protein that is composed of extracellular domain, transmembrane region, and cytoplasmic tail. And the unique feature of CD44 is that it is composed of 10 variable exons that are located in the extracellular domain. And including these variable exons results in generation of peptides that can interact with growth factors such as FGF and HGF, leading to formation of co-receptor complexes with receptor tyrosine kinases that results in activation of cellular signaling inside cells. In addition, these variable exon regions can also result in a peptide generation that can interact with proteases such as MMP7 and MMP9. It can also interact with the proform of TGF-beta for the maturation of TGF-beta, which again is a quite important cytokine that controls cellular processes. In contrast, cells can also exclude all of these variable regions, resulting in the formation of another isoform of CD44 called CD44 standard form. This isoform is known to be involved in cell-cell contact inhibition. So this isoform, through interacting with growth factors and receptor tyrosine kinases, it activates signaling inside cells, leading to cell proliferation. However, this isoform is involved in cell-cell contact inhibition. So again, by regulating alternative splicing, cells can control whether they want to proliferate or they want to stay in a non-proliferative state. It is known that different isoforms of CD44 are involved in different functions inside cells, and that are involved in the regulation of cell growth, cell adhesion, migration, and invasion. And again, regulation of isoforms of CD44 as an example is quite important in controlling cellular processes. So showing here is just a schematic of the pre-mRNA structure of the CD44. Again, these gray boxes are constitutive exons that are included in every single case of the final message RNAs. The magenta ones are the variable exons that can be included and can be excluded. And you probably noticed that inclusion of these variable exons are quite variable. So what that means, variable, what that means is that you can have all of the exons, variable exons included, you can have a single variable exons included, and you can have multiple different types of exons included. And these are also regulated by cells. And different isoforms of variable CD44 or CD44 variants also play different roles, which I indicate over here. So it has been shown that inclusion of the CD44 variable 3 exon is involved in recruitment of proteases such as MMPs I just mentioned to you for the maturation of growth factors. Inclusion of the variable 6 exon can make these CD44 variants to act as a co-receptor with HGF and MET. Those are growth factors and receptor tyrosine kinases that lead to activation of cellular signaling inside cells. And it is also shown that inclusion of the variable 6 and variable 7 exons are important in terms of inflammation. So what we have shown is that as what has been asked, whether this is regulated, yes. And what we have shown is that in addition that alternative splicing is regulated, isoforms of alternative spliced variants are also important for feeding back to this regulation. And shown here is a positive feedback loop that couples Ras/MAP kinase signaling, shown here, and alternative splicing of CD44. So again, this is the cellular membrane, and CD44 is the cell membrane protein that can, this variable exon V6 containing variant can form co-receptor complexes with growth factors and receptor tyrosine kinases. This co-receptor activates Ras/MAP kinase signaling inside cells. And activation of Ras/MAP kinase signaling can further stimulate alternative splicing of CD44, resulting in a generation of CD44 splice variants. And these CD44 splice variants are synthesized as proteins, resulting in the presence of these CD44 variants on the cell membrane. And again, these newly synthesized CD44 isoforms can further form co-receptor complexes with growth factors and receptor tyrosine kinases, leading to sustained Ras/MAP kinase signaling. Why I say sustained? What that means is that as long as you have growth factors in the vicinity of the cells, cells will keep Ras/MAP kinase signaling on. And Ras/MAP kinase signaling is quite important for cell proliferation. And what that means is that cells now stay on a state where it is proliferative. It is growing. However, previously, it can be a non-proliferative stage, meaning that it can be quiescent. So through regulation of alternative splicing, one can switch a cell type from a quiescent state to a proliferative state. And you can envision that cancer cells are those that are proliferative rather than quiescent. And again, what that means is that if you upregulate the level of alternative splicing of CD44, you can convert a normal cell to a cancerous cell. And in fact, it has been shown that many of the CD44 variants are upregulated in cancers. And shown here are just a few examples that has been published showing that in many types of human tumors, different isoforms of CD44 are upregulated. And our lab is using this model system and to understand how CD44 alternative splicing is regulated and how this regulation pertains to tumor genesis. So how is this regulated? Now, mRNAs are made by recognition of a few splice sites called 5' donor splice site and 3' acceptor splice site. And it's GU and AG. And in between, there's also a branch point. So what happens is that there are multiple proteins involved in splicing, and those are in the spliceosome. And the spliceosome can recognize this 5' donor site as well as 3' acceptor site, resulting in the joining of the exons. So these are constitutive exons. In terms of the variable exons, they often have very weak acceptor site and donor sites. So these sites require help for the recognition by spliceosome. And what happens is that in many of the variable exons, it contains exon splicing enhancers, shown in green, or exon splicing inhibitors. Yes. So, as those sequences can be located in the intron regions. There are a group of proteins such as HNRNPs that recognize the silencer regions of the sequences in the introns or in the variable exons, resulting in the inhibition of inclusion of this variable exon. So that promotes constitutive splicing. However, there are also a group of proteins, such as SR proteins, that often bind to the exon splicing enhancer sites that results in the recognition of these weak splice sites, leading to the inclusion of the variable exon, which means that it promotes alternative splicing. So in conclusion for this part, what I have told you is that in higher eukaryotes, such as mammals, one DNA can generate multiple isoforms of mRNA through alternative splicing. And these mRNAs are translated into different isoforms of proteins, and in many cases, these proteins have different functions. And this results in a complexity in humans. And this process requires tightly regulation, and dysregulation of this process results in diseases. Now, I'm going to switch gears to talk another level of post-transcriptional regulation, which is at the level of microRNA regulation. So what are microRNAs? MicroRNAs are small non-coding RNA molecules that regulate eukaryotic gene expression post-transcriptionally. And often the processed microRNAs that are functioning are only 20-some nucleotides, very short. So what they do is they form a complex with RISC. Those are proteins, and that target the untranslated region at the 3' end. It's called 3' UTR, that can inhibit translation of a protein. So microRNAs can have quite a few different modes of actions. And in mammals, in general, microRNAs are involved in translational regulation. What that means is that it recognizes a region at the 3' UTR, and it forms a complex with RISC, and that stalls the translation process. So these are the ribosomes. So proteins cannot be made, although messages are made. In addition, it has been found that in plants, as well as lower eukaryotes, microRNAs can act to degrade mRNAs. So they can again find the site where where it can base pair to so form a complex, and there are scissors such as the RISC complex can chop the mRNAs so that mRNAs are degraded. Furthermore, in lower eukaryotes, such as yeast, it has been found that microRNAs can be used at the level at the transcriptional regulation. So it can again form complexes with RISC at the promoter region of a gene. And this results in the recruitment of histone modification enzymes, leading to histone methylation, resulting in the silence of that gene. So microRNAs can shut down the transcription of a gene. However, as I just mentioned to you, in mammals, in general, translational regulation is a key. So where is microRNAs made? microRNAs first is encoded in the genome, and it's non-coding mRNAs, and it is often folded as hairpins, as I shown here. So in the nucleus, when microRNAs are transcribed, it's folded, and there's an endonuclease called drawsher can process microRNA to pre-microRNA. And after this processing, there's another protein called X protein can transport this pre-microRNA to cytoplasm where dicer is in. Dicer is another enzyme, endonuclease enzyme, that further chops microRNA to short RNAs, and these are about 23 nucleotides long. And these microRNAs are recognized by RISC complex, further find their their target sites, such as the 3' UTR of a gene, resulting in the inhibition of translation. And sometimes microRNAs can be involved in cleavage of mRNAs. And again, these are often found in the lower eukaryotes. So microRNAs are often conserved. And shown here are just a few examples going from C. elegans to humans. And this is miR1, microRNA is 1. And you can see that the red are the regions that will find the targets, and you can see that the nucleotides are quite conserved among these 4 species. Although the loops over here, as well as other regions, do not need to be conserved. So microRNAs are also found to be dysregulated in cancers, and microRNAs can act on both tumor suppressor genes as well as oncogenes. So for example, here's an example using miR34, microRNA 34 as an example. And P53 can transcriptionally upregulate the expression level of miR34. And miR34 can act on oncogenes, such as the cyclin E2 CDK complexes, which is involved in cell cycle progression. And miR34 can also act on the BCL2 protein, which is involved in cell survival or apoptosis. So this complex is involved in cell cycle proliferation. And miR34 inhibits the synthesis of this complex, leading to inhibition of cell proliferation. In addition, BCL2 is involved in inhibition of apoptosis. miR34 inhibits BCL2, and the net result is that miR34 inhibits the survival of cells. So what that means is that miR34 is a microRNA that inhibits oncogenes. And there are also other microRNAs that inhibit tumor suppressor genes, and those are called oncogenic microRNAs. And I've shown you here a few examples. And for example, some of these microRNAs inhibits the translation of E2F1, and leading to further processes of inside cells. So nowadays, many people have thought of using microRNAs as a tool for the treatment of diseases such as cancer. And for example, one can deliver microRNAs into cells to kill cancer cells. And for that, there's a term called RNAI, or RNA interference, meaning that you can synthesize a short RNA and then deliver it into cells. And what I will tell you next is RNA interference. Probably some of you have heard. So RNA interference can be used as a therapeutic tool as well as a tool for biochemical studies. So, oops. So RNA interference is also called RNAI. It is a way to silence gene expression. And often you introduce double-stranded RNA into cells, and then this double-stranded RNA can degrade, can first find their targets with high sequence homology, and then degrade those mRNAs. RNAi can be achieved by several ways. For example, you can introduce double-stranded RNA that you synthesize. You can also introduce short interfering RNAs. Those are 21 nucleotide long duplex RNAs, also called siRNAs. Many people now use this in the laboratories. And you can also use short hairpin RNAs called shRNAs. So for double-stranded RNA, as well as shRNAs, they are processed in cells to make siRNAs. And siRNAs are made outside of cells, so for example, by us. And then we introduce these already processed siRNAs into cells so that they can readily find their target for silencing the gene. So siRNAs has a defined structure. It has this duplex region, and it has 2 overhangs at the 3' end. And this duplex region is about 19 nucleotide long. And it is this antisense strand that acts as an active strand to be involved in the risk complex formation, and then further targets to find the targets in the cell. So the way that siRNA works is the following. So you can, for example, introduce this siRNA into cells, and then it finds the homologous mRNA, and this siRNA then form a complex with risk, and leading to the cleavage of these mRNAs, resulting in the degradation of that mRNA, so there's no protein that's made. And for this, it's at the level of mRNA degradation. And it's different from the microRNAs inside cells because we are introducing these siRNAs in cells. And also, the difference is that for siRNAs, you have to have almost like 100% base pairing. For microRNAs, often you form bulges, meaning that there's a region that is not base paired with the target mRNA. Now, it's nice to have siRNAs. However, it also has disadvantages. For example, it's transient. So for those who do transfections inside cells, after a few days, this siRNA effect will be gone because when cell divides, the siRNAs will be diluted. So for example, if you have 100 siRNA in one cell for day 1, day 2, when cells are divided into 2, you only have 50 siRNAs. And day 3, you only have 25, and so on. Another thing is that some of the cells are very hard to be transfected. And in that case, you cannot introduce siRNA inside cells. And that made researchers to develop shRNAs. For example, this is just one of the examples of shRNAs where it has this mere microRNA structure, and it also contains the regions that you are interested in, for example, the red region. So this is a region, for example, if you wanted to target a gene of interest, let's just say CD44. So you put the sequence of CD44 over here, and then this is encoded in a plasmid. So plasmid is infected into cells. And then this plasmid can be integrated into the genome of that cell, and this region is transcribed. And further, inside cells, there are machineries that can process these microRNAs, the pseudo microRNAs, results in the generation of siRNAs, just like if you can transfect the ready-made siRNA inside cells. So as I just mentioned to you, siRNAs can be delivered inside cells, and there are a few starting points, that the materials that you can use for the silencing of a gene that you are interested. For example, you can synthesize a long double-stranded RNA, and you can use a tube to add dicer complex in. So biochemically, you can process those long double-stranded RNA into siRNAs and then transfect into cells. Or you can micro-inject those double-stranded RNA into cells, and the cellular machinery of dicer can process this long double-stranded RNA, making the short siRNAs. And for the shRNAs, it can be made using a plasmid-based shRNA, like the transfection, as well as the infection. And in that case, through generation of a microRNA structure, as I shown here, it can be processed and with the generation of siRNAs. And the final product of siRNA can be used to silence the gene of interest. So the short hairpin vectors has been used quite widely nowadays, both in the laboratory as well as in a company, in terms of trying to develop tools for therapeutic cues. For example, as I said to you just now, it can be used in laboratory for infection of tissue culture with the generation of the shRNAs or inside cells, which can be used for high throughput analysis, as well as if you're just interested in one gene, you can also knock that gene down, the expression of that gene down. It can also be used for knocking down a gene inside animals, such as a mouse. For example, you can introduce the shRNA into ES cells and making a mouse line, and you can let that shRNA express in that mouse and see whether if you knock down that gene, would you be able to see a phenotype. And the final goal is to use the shRNA technique to see whether we can achieve any therapeutic outcomes. So the conclusion of this second part is that microRNAs are the small non-coding RNAs that are processed inside cells, and microRNAs can be functioning as both tumor suppressor genes as well as oncogenes. And this process, again, needs to be highly regulated, and dysregulation of microRNAs can, again, lead to changes in cellular states, or such as cell cycle progression, as well as apoptosis, leading to the development of diseases such as cancer. So I will end here, and if you have any questions, I'm happy to answer them. Thank you. Yes.
So most of the clinical therapeutic application, they try to use microRNA or siRNA based. I'm just wondering if there's any therapeutics attempt they try to targeting this alternative splicing, and if so, how can they achieve this?
Sure. Yes. So it's a very good question. Yes, in the first part, I mentioned to you that different isoforms of a protein may function differently. And the goal in my lab, in fact, is to prove that it's true in human cancers. And if that's true, then one can target alternative spliced isoforms. The way to do that is that you can target those variable exons rather than the constitutive exons. So variable, for example, let's just see variable 6 exon containing CD44 is involved in cell proliferation. And what you can do is to design siRNA or shRNAs to target that variable 6 exon, so that only variable 6 exon containing CD44 isoforms will be degraded. Other isoforms that do not contain variable 6 exon will not be affected. And in fact, that has been shown by my lab, and we have also shown that only targeting variable 6 exon can affect cell proliferation.
My second question is, how do you define some siRNA sequence that have high homology to So that's about the off-target effect. So when you design siRNA, how high, what's the, I don't know, percentage of homology do you consider to be high?
So for designing siRNAs, often people do 100% base match, meaning that for that 21 nucleotides or 19 nucleotides, you will find exactly the match with the gene of your interest. And there are different, so yes, there are off-target effects in cells. That's why you want to use not only one siRNA, but many times 2 or 3 siRNAs targeting the same gene, but at different locations and show the same effect. In addition, what you can do is you can knock down that gene by siRNAs and reintroduce the gene back using siRNA refractory cDNAs of that gene and see if you can rescue that phenotype.
Like most of the microRNAs have like variety of targets, and say if we just knock down a microRNA which just functions as an oncogene, whether it will affect its normal effect. So I mean, like the gene therapy sometimes may bring some like side effects. Say like the microRNA may not just target this target, right? Thanks.
Yeah, so the question is, microRNA often have multiple targets rather than just one target. What about if this microRNA targets one oncogene, however, it also targets some other genes that are quite important? So if you get rid of this microRNA, although you kill tumor cells, but you probably will kill the normal cells as well. That's, in fact, is a question that people have. So when people identify a microRNA that can be potentially used as a therapeutic strategy, they always want to make sure that this microRNA doesn't really kill the normal cell, but only target the tumor cell. And it's a practical question as well. Thank you.